Okay, we're back. I'm going to kind of finish up that basic part by saying, as far as your hive setup is concerned, we like to point our entrances kind of southeast in this area. It gives us a nice little southeasterly breeze to kind of keeps the hive cool, and also serves as a way to get that north wind at their back in the wintertime. So we can maybe even put a wind block up of some sort. Some guys use a big bale of hay or a couple of panels of sheets of plywood or whatever it is. If you're going to have a really bad windstorm, it's nice to have a little protection. It helps the hive stay a little bit warmer. Honeybees have a really cool ability to unhinge the wings from the muscles that they use to fly with, and they cluster in a cluster, and they vibrate. They shiver those wing muscles like they were flying, and it creates a heat, and it keeps the cluster warm. And the bees on the inside will cycle out and let the ones on the outside in to warm up, and so forth. The brood nest, as we'll talk more about, it looks something like this. You're going to have a section of your hive that's full of eggs and larvae, and they have to keep this nest approximately 94 degrees, 95 degrees. 93 to 95 is what the science says. If it gets cooler than that, the brood may chill. If it gets warmer than that, then it doesn't develop properly. Our honeybees can stand a hotter temperature, but the nest and the eggs and larvae cannot. In fact, the bees will heat up other insects. They will cluster around them. They will ball them in a ball and vibrate and shiver and create this heat, and it's like a little miniature oven. And they can even kill a queen like that. That's one of the ways they kill foreign queens. If a foreign invader comes in, or say a swarm moves in, that swarm may try to kill your queen or the existing host queen by doing the same thing. They cook them. They get that temperature up higher than that queen or that other insect. A wasp of some sort cannot handle the same temperature as a honeybee. So that's one of their defense mechanisms. Uh, but with that said, I want to start just to share with you one of the other things as far as setting up your hives. A little afternoon shade is not a bad thing in August, unless you're having a hive beetle problem. Hive beetles is a little black bug that looks kind of like a little dome shade, almost like a larger regular beetle, but just a little bitty dude. And they want to lay their eggs in your honey. And if given the opportunity, they will. Your bees will keep them in check. A strong colony will keep them in check for the most part. If they ever become a real nuisance, there's a couple of options. They sell a beetle trap like this. We sell them. We have them. They're about $1 a piece, I think. You put a tiny bit of oil in the bottom of this. It's got holes in the top that the beetles can go in there to hide from the bees and get away from the honeybees that are chasing them. And they get caught in that oil. Now they can't get out, and they drown and die. There's also some cloth. It's like it's kind of like a dryer sheet. Some people use dryer sheets to try those, and bees will frizz those, chew them, and kind of get them fuzzy, and it serves like a Velcro. The bees don't do it so the beetles get caught, but the bees inadvertently get caught in those cloth traps and get stuck and can't get out. Beetles are not normally a problem. Wax moths can be a problem if you have a weak colony. If for any reason your hive goes weak, lots of things are going to get in there and start to cause problems. Wax moths are probably the most common. The hive beetles as well, and then you'll have occasional intruders like there's a couple of assassin bugs that come around and get into beehives. Lizards might hang out and catch one or two. I wouldn't be alarmed about that. Most of the bugs and things that come around and catch a bee on the outside, or spiders or whatever, they're not going to catch enough to hurt anything. They're not going to do any damage to your colony. In other words, again, your queen's laying about 2,000 eggs a day. That's what I want to talk about next, is the life cycle of the different cast of members of the hive. Queens are going to go about 16 days from egg to emerging as an adult. When the queen lays an egg in a cell, she measures the cell, the depth and the width, to determine if that cell capacity will hold a drone. If it does, she turns around and deposits that egg and does not fertilize it. She checks it if it's the size that will house a worker bee. She turns around, deposits the egg, and she fertilizes it as she lays that egg. She fertilizes each one individually. They don't come out pre-fertilized. And it's based on the size of the cell that they're going to develop in as to whether she fertilizes or not. The worker bees, they take about 20 days to go from egg to emerging as an adult. A lot of the books say 21 days, according to some folks like Randy Oliver, who have actually done some some tests and trials to see, and marked and checked them. 21 days, 20 days, somewhere in there. So anyway, depending on a few variables, possible weather conditions and such. The worker bees are going to be the bulk of your hive. They're also going to have approximately 5% drone cells, and depending on what time of year it is. They don't raise drones in the winter. In fact, they kick them out. They don't want to feed drones in the winter. They don't mind feeding drones in the spring and summer when there's mating of other queens going on, because they want to pass on their genes. But they eliminate them in the winter because there's not much breeding activity, and they don't want to feed. It's a waste of food in the colony's mind to support drones throughout the cold time and the lean times. So they'll kick them out. But the drones take about 24 to 25 days to emerge as adults, or to hatch, if you will. It's important to know that for a couple of reasons. One is to do with the varroa mites. If varroa mites have their choice, they will go into a drone cell, and because they know they get a couple extra days to lay eggs, that's how they reproduce, is inside the cells. We'll talk more about those later. This is what it looks like when a queen goes out to get mated. Hopefully you can see that okay. Each mating session will last a few minutes, and it'll consist of, like I said before, 15 to 20 males. 20 drones will mate with the queen in one mating flight. Each one will pull the parts from the one before. When the male finishes his mate, he falls back, and the next male pulls his parts out. He falls to the ground and dies. So it's maybe not the best idea to be a drone bee when you find the queen. Then that's pretty much the end of the party for you. But they hang out in what's called drone congregation areas, and they've done a lot of research on this, how that works, and how the different drones of different species work, or different families of bees, or different altitudes. They found them, and some fly at a little higher or lower heights than others. Queens are going to go a couple of miles from their home colony, so that they're not interbreeding with their own younger or siblings or offspring. I'm sorry. They'll travel a couple of miles away, and they'll mate with drones from that area. So if I'm going to try to raise my own queens, which we do, we surround our mating yards within a couple of miles of our good, strong, good genetics, and I flood that area with good quality genetics, so that I know we're not getting a bunch of possible Africanized genetics into our genes of our queens. A lot of people discourage people from raising their own queens in Texas because of the gene pool is in our wild bees, or our even some managed hives in this area. They're not necessarily what you would want in your breeding program. So it takes a lot of hives to surround your mating yards with, to guarantee your gene pool is going to be strong. I used to hear 200 was a good number, but I like to have at least 400 or 500 hives that have good genetics of good drones in our mating areas. So I'll surround up our mating yard in every direction for 3 or 4 miles, or 2 or 3 miles, and that way the drones can be there to service those queens that go out and get mated. Like I said, once the queen's mated, she comes back, she's full, she's done for life. She's not going to fly again. She's never going to leave the hive again unless she swarms. If she takes off with a swarm, then of course that's reproduction, and that's where she goes and lands in her new home. She'll stay there. She's not going to go get mated again, in other words. When she lays the egg, it's a little tiny thing. It's a very, very tiny speck in the bottom of that cell. It's very hard to see. When you're first learning about bees, that's one of the first things you're going to want to learn how to do, is find eggs and spot eggs. A lot of people get caught up in whether they saw their queen or not. Was the queen marked? Is she marked? Oh my gosh, my queen needs to be marked so I can find her. I don't necessarily think that's a big deal. I don't look for my queen every time I inspect a hive. I want to go in there and check and see if I see evidence of the queen. And when I say evidence, if I see young larvae, if I see these little white guys, then I know I've had a queen in that hive in the last few days, because these, after about a week, they put a cap over them, and they cap them, and then the baby bee will go into the pupal stage, and it's like a cocoon for a butterfly. It develops into a larger bee inside this cocoon for about 2 weeks after they cap it. So that 20 to 21 day window, it starts off as an egg for 3 days, it hatches. It forms, after it hatches, it lays down in the cell, and it forms kind of a banana shape, and very quickly turns to what's called a C-shaped larva. The C-shaped larva will consume food. The bees are going to feed it royal jelly all day, every day, for its entire growth cycle through its 5 instars, and it fills up that cell very rapidly, and within a couple of days, that little white grub is filled it up, and it's standing up. It can't eat anymore. I mean, it's just filled itself up, and it would be the equivalent of us in 7 days eating so much we turn into the size of an elephant. I mean, that's how fast they grow. And when they fill it up, that fifth instar is when they cap this cell and the little baby emerges. Well, if I see these guys, I know my queen's been in there within the last week. Pretty good chance she didn't swarm, unless I see a bunch of swarm cells. And I've got some pictures I'll share. I'll probably try to incorporate them into this video if I can figure out how to do that. This is kind of a new toy for me, so I'm going to have to look into that. But at the very least, I'll make sure that they're available. There's some on our website already, and I'll make sure and create a page on our education section that points these out, because you need to know what swarm cells look like. A swarm cell is basically a cell that the hive has created for a queen to develop in, because they've got the urge to swarm and reproduce, and they want to lay eggs for the colony to raise a new queen, so that when the old queen leaves with 3/4 of the bees, these guys can raise a new queen that inherits all of the young, all the wax, all the larvae, all the food stores and everything, and she'll go out and get mated and come back and start again. So again, if I can see sign of the queen, an eggs tells me she's been there within 3 days. These older larvae tell me 4, 5, 6, 7 days. I'm satisfied. I put this back in and I go to the next hive. I also, while I'm checking my hives, I want to look for food and make sure we've got plenty of food and also plenty of room for the queen to lay more eggs. If the gap between the brood nest and the food rainbow, which I'll point out later and show you when we do the open hive section of our class later when you pick up your bees, when that section of the nest grows together, that eliminates a place for her to lay more eggs. There's a scent that is associated with the young brood. Some people call it young brood scent or young brood pheromone. It's ebaosmine is the scientific name for it. And that scent tells the colony that we have young brood and everything is happy, everything is healthy, we have a laying queen, everything is great, all is well in our world. When that scent is no longer available because the queen has run out of room to lay eggs, and all of those young bees, the young larvae have grown past that stage, and then there's no young ones to replace her, that's an indication they've filled up the cavity, they've filled up the tree space, and there's no more room to lay, so it's time to reproduce, and that's when they start producing swarm cells and get the urge to swarm and take off and leave. And again, you don't want $200 and some odd worth of bees to fly off, so we give them more space. We add another box, or we can do what we call a split or a divide. We can take partial parts of the frames out. You don't even have to do a complete split. You could just take a few frames out and share them with a weak colony, perhaps, or you can start a new colony and you can put a couple of blank frames in and give the queen more places to lay. As soon as it gets more of the ebaosmine back in the colony, the swarm urge is reduced. That's just one of the triggers, but that's one of the main triggers. Again, you need to understand the life cycle for a few different reasons. One has to do with the mites and their life cycle and how they reproduce, but the other is the awareness of how long it takes these things to happen makes more sense of things in terms of what goes on if your colony does swarm, for example, if they're going to raise their own queen. A lot of people say, well, can't I just let them make their own queen? Well, sure you could. Again, you don't know what the gene pool is like where she's going to go get mated unless you're out here by us and we have several hundred hives. But the other thing is the success rate is just not that great on queens going out and getting mated and come back. 75, maybe 80% on good conditions. There's lots of things that can cause that number to not be 100%. There's Mr. Mockingbird or Mrs. Mockingbird that has babies to feed. If they spot a queen, she's a lot bulkier, easier to see, maybe not as nimble on the wing. Maybe a mockingbird or something else that really catches a lot of bugs is dragonflies. Don't kill dragonflies. Unless you, if you're a queen breeder, it's one of your biggest enemies. But if you're in a place where you have a lot of mosquitoes, dragonflies will help your mosquito population. A lot of people say those martins are good, but the dragonfly population really helps it. But I digress. Having your colony raise their own queen is risky because let's say that she goes out and gets mated the day one and comes back and she gets the urge to go out again. Maybe it's raining like crazy, or maybe she never gets to go out and get mated because of the weather. Her mating flights get interrupted by weather for 3 or 4 days, and then when it does clear up, she goes out and who knows, a storm could blow up, windstorm, rainstorm, or she could simply come back and not find the correct box if there's multiple boxes around, multiple hives around. So there's a lot of things that cause queens not to get back to a hive successfully. I would say if you have a hive that's swarmed or is about to swarm, don't kill all the swarm cells automatically, even though that's not your first choice. See if you can locate a queen. Call me or get online and look at some of the other websites that's people that sell queens. If you can get a replacement queen, then go ahead and knock out the swarm cells and then get your new queen. You want it to be queenless for at least 24 hours before you introduce a new queen. That gives the colony time to realize that they're queenless. We have either queen right or queenless colonies. And a queenless colonies will accept a new queen, but you don't want to just stick her in there right away. We have introduction cages, we call them. I'll show you what that looks like. An introduction queen cage would look something like this. There's different types and styles, but there's a screen window where the bees can feed and get to know this queen and exchange pheromones with her while they're waiting for her to come release. You can manually release her. There's a cork in one end of this. One of the things we do is we put a plug in the end that has got what we call queen candy in it. This candy is basically a mix of sugar, powdered sugar and honey or syrup, and it goes into one end, and it's got enough candy in it that it takes the bees about a day or two to eat that much candy. The bees in the colony, not the queen. They call it queen candy, but it's because it's used in queen cages. You slide this in, you pull the cork out, slide this in, it keeps the queen from getting out. Put it in the colony, and in a couple of days they'll eat this candy, then she can crawl out. It's like a timed release mechanism for the queen. When she comes out, by the time that she gets released, usually their scents are acclimated and the success rate is very high. A colony that you just put it in may or may not accept her immediately, so we do the slow introduction by this method. Some queen breeders ship queens with attendant bees in the cage with the queens, and some send them with a box with the queens on the attendants on the outside of the queen cage. I prefer them on the outside, but that's just my own style. As far as the other things, this is what a swarm is going to look like when it lands in a temporary place. They're very docile. They're not aggressive at all. You can walk up and basically put your hand through it. I go through there and I'll move them around looking for the queen. I find her, put her in a little hair clip queen catcher, or one of my little tubes or whatever I use to hold her with. Now I can put this in a box and they'll all come with her. Shake this into a box if it's on a small enough limb. I've seen these on everything you can think of. In fact, I've got a friend that's a police officer in San Antonio. He sent me a picture of a swarm of bees on the side of a police officer's car, and they got a big kick out of that. But this is what it looks like in flight. They're basically a cloud of bees. You may have seen or heard of them. You can hear them. They're kind of loud. You can look up and see the air is kind of full of bees. And that's what it is. It's a swarm. And they're basically looking for a place. They're going to send out scout bees. There's a lot of information. There's a great study done by Dr. Tom Seeley. He took a bunch of bees out to the island and did some studies on swarms and the size cavities they like in that. I won't spend the time with that. I encourage you to look up and Google Tom Seeley, Randy Oliver, and Jamie Ellis is another good one. I love to hear Dr. Laura Rangel, our own Texas A&M Bee Lab. She's head of our Bee Lab. She's great. She's fun to listen to. She has history with Africanized bees. She's from South America, so she can tell you a lot about that. Just her lectures, or Google or YouTube. Go on YouTube and look up the National Honey Conferences. They've got a lot of great speakers that come on those. You can learn a lot from those guys. I say follow the trusted sources for your bee research. It's a lot of fun to read stuff from Billy Bob's bee blog. Don't get me wrong, Billy Bob might have better bees than me and you both put together, but Billy Bob may or may not update his blog. He may not have the most current, up-to-date information on mite treatments, etc., etc., etc. So just watch the source. If it says .com, like mine says .com, thebeeplace.com, that means I'm probably selling something. And that don't mean you can't trust me. It just means that I'm not a research lab. I'm not a research facility that while I do have a large lab, I have over 1,500 hives. I have a lot of bees that I can study, but I'm not accredited laboratory. I do not have the credentials that goes with the serious research that's done by folks like Randy Oliver and Jamie Ellis and the big guys. I help them. I work with them. I share research with any of them. Texas, I'm part of, like I said, the Master Beekeeper program, so I share information with the folks at Texas A&M and Randy Oliver. There's several mite treatments that he's worked on, and he's responsible for getting a lot of the medications and things approved for the beekeeping industry because he has the background as a research biologist and a scientist, and he has the credentials. He has a large enough lab that he can take 25 or 75 hives, isolate them in a quarantine yard, and he'll throw a virus at them just to see what'll cure it or what won't cure it, or what's good or what's bad. He'll run the trials and turn these papers into the USDA and get things approved as needed. In fact, he's a regular. He writes an article regular every month in American Bee Journal. Anytime I have a serious question about honeybees, I mean, I look for people like him to help me come up with the answers. Normally, the bees will tell you what you need to know, but there are things that even some of the experts scratch their heads about. Again, 500 years from now, we might have it figured out. Meanwhile, we enjoy the journey. We enjoy learning together. I think it's really important to understand that it's a fun journey. It's kind of like a kid. You know, you get a new bike at Christmas, and your brother gets one too, or your cousin got one, or your neighbor got one, and you want to be the first one to learn how to ride a wheelie or whatever. And you're both probably going to skin your knees, and you're going to both come home with a scrape or a scratch, and somebody may even get a broken leg or a broken arm before the summer's over. But at the same time, you're having fun, you're learning, and it's a fun thing. It's a fun journey. Beekeeping is similar in the sense that it's exciting, it's fun, it's fresh, it's new, and there's a lot of area unexplored. In fact, science admits that they'll never know all there is to know about honeybees. They're just that extensive and that fascinating, and they're great. With that said, I want to get this closed and start to the next section. We'll talk about the varroa mites. Again, if you have questions, jot them down. If I don't cover it in the next segment, then we'll try to do a live session where we can answer all the questions you have.